Welcome to Hungarian Horizon, a podcast about Hungary, its people, and their stories. My name is Balázs Frey.
And I'm Barbara Bálint.
Here with us today is Dr. István Peták, an internationally renowned cancer researcher and pioneer of predictive molecular pharmaco-diagnostics for anti-cancer targeted therapies. Dr. Peták is the co-founder of Oncompass Medicine, whose software solution for helping doctors find the best targeted medication based on identified genetic defects won Digitaleurope's 2021 Future Unicorn Award as the most promising technology startup in Europe. It's an honor to have you.
Thank you very much. It's a pleasure to be with you today and talk about our research and company. I'm very excited to share our thoughts.
Thank you.
Thank you for joining us today. What made you decide to want to become a cancer researcher?
So as a teenager, I went to the woods and forests together with my father, who was a very well-known backpacker and naturalist who promoted going to the nature. And so I fell in love with the animals and plants, and so I went to do volunteer work into a national park where we had to save animals from diseases. So, but then I realized that with the knowledge of, with the power of knowledge, humans can save lives, and I felt that is the most beautiful thing you can ever imagine. And then I thought that because humans are part of nature, and if we understand how nature works, and we understand what's the harmony in nature, we can also understand the cause of diseases, and we can cure them based on our understanding. So this made me go to the medical school, and then the last year, I had some research assignments in cancer research, and I chose cancer as one of the most significant diseases humanity has to face for thousands of years, and it's actually becoming the number one killer of humans. It's actually every minute as we speak now, 20 people die in the world. So it's we are under extreme pressure to find a solution. But then when we realize what the cause is, we can focusing on to attack the cause and solve the cause. And so in case of cancer, we discovered in 1960 the first cancer gene. So that's the first proof that actually cancer is caused by genetic alterations. And since then, we have been doing research how to fight cancer on the level of the cause. And the breakthrough came in 2000 when this first alteration, which actually causes chronic myeloid leukemia, it's a translocation of two chromosomes, and which creates a fusion protein which accelerates. It gives the order to the cell to divide and grow, but in 2000 there was a brave scientist who used the targeted therapies on the first time to target this mutant gene, and we had a breakthrough, and now most patients with this type of disease can be cured or kept alive for years. Now later we learned that unfortunately cancers are usually heterogeneous on the level of molecular alteration. So there are more than one genetic alteration which can cause cancer. Actually, right now we know the exact number, almost the exact number. It's 6 million mutations in 600 cancer genes. And each patient has a combination of 4 or 5 mutations in each cancer. So we are facing extreme complexity. So each cancer gene can cause multiple type of cancers, and each cancer can be caused by multiple different type of mutations. So if you want to fight cancer with the right treatment, we have to know the exact molecular profile of the patient's tumor, which mutations are present, and we have to choose an individualized, personalized therapy. And today we have more than 100 targeted therapies already in clinical use, which is great. So, which enabled us actually to reduce the mortality of cancer by 10% total. And in some patients, we have great breakthrough effects. But the challenge is today that we have to do a very detailed molecular profile, molecular diagnostic test for each cancer patient. And then, based on the individual profile, we have to predict which therapy will work. And it's an enormous challenge. And this is something where we are now. We try to improve our diagnostics, and we have to develop more drugs, and we have to develop tools which help doctors to choose the right therapy.
So does this mean that we can be optimistic about finding the treatment and cure of cancer?
Yes, I think we absolutely must, and we can, simply because we see that it's possible to stop cancer with these targeted therapies. Mm-hmm. So I think the next 10 years we will have another 200, so we will have in 10 years 300 targeted therapies that will cover like 70-80% of cancer patients. So it's just mathematics now, and there are some technologies which we develop, which we hope that will accelerate this development.
And so this matching of type of cancer to treatment is what you call predictive molecular pharmaco-diagnostics for anti-cancer targeted therapies, or is that something separate from this?
Yes, and sorry for these terminologies. Actually, we call this whole approach precision oncology today. When you give an individualized targeted therapy for a cancer patient based on the individual molecular profile, the specific combination of mutation the tumor harbors, and yes, the whole idea was at the beginning to develop diagnostic tests which predict the response of a drug to the drug. Of course, just to make sure that we understand that as a doctor I have to tell that if you have a cancer which you diagnosed as an early stage, surgery is the best solution usually, and you can be cured. So that's why screening is extremely important. Now, what we are talking about is the metastatic cancer, when the cancer cells spread all over the body, so it's not possible to take them out. So you need drugs, medicine. So in 2003, when I came back from the US, I met one of my classmates, Richard Schwab, who was coming back from Basel, University of Basel. He was doing research on targeted therapies, and I was doing research on signaling pathways and cancer genes. So we put it together to found this company, and we first focused on developing single gene diagnostic tests. And this is how we discovered the first target, the first gene in lung cancer, and actually organized the first prospective treatment of a lung cancer patient based on the genetic alteration causing her tumor, and we published it in a major medical journal of the Journal of Clinical Oncology, the Journal of American Oncologists, and so we were pioneering this field, and then but we saw that this mutation only occurs in 5%, so we had to develop novel methods which cover all possible mutations in cancer patients. So in 2008, we started to develop multi-gene assays, or molecular profile assay, based on a new technology called next-generation sequencing. Sorry for this, but we have to learn it, I think, because it's like microscope. Everybody knows what microscope is. It's the same thing with the next-generation sequencing, or NGS, because it's the molecular microscope. With this method, you can look into the cell and make a molecular X-ray of the whole genome and see all the mutations or what caused the cancer in that patient. So that was our second focus. And 5 years ago, we realized that, okay, it's great. Now we can see all the mutations in the patient, but we realized that the complexity overcomes the ability of the human brain to combine all the information about all these millions of possible combinations of mutations. And the next hurdle will be how to predict whether a drug targeting a single target will be able to stop a cancer which is driven by multiple genetic alterations. Mm-hmm. Sometimes it's possible, sometimes it's not. It's like when you have a building you want to demolish, and it stands on 5 pillars, and you have 1 kilogram dynamite. You want to know where to put it. Mm-hmm. Because if you choose the right spot, it's gonna be enough, but if not, it's not going to be enough. So we realized that that's the next problem we have to solve, so we started to develop a method to solve this, and this is the computational method which we developed and deputed in the last year.
Okay. So how do you imagine Oncompass Medicine will grow in the next 5 or 10 years?
So we have been awarded the Future Unicorn Award by Digitaleurope. So it's a great honor. It's a very prestigious organization of 35,000 companies in Europe in 40 countries. Wow. And these organizations selected us as the future unicorn. And the future unicorn means in the startup world a company which worth of $1 billion. So we have to live up to this expectation.
It's a high bar.
Which is great. But I think this will translate also to millions of lives we can save. And I think this is the challenge. And so to achieve both, we have to go to the international market and to present in as many countries as possible.
We wish you the very best of fortunes in living up to your momentous promise, and hope to see you on the front page of Time magazine sometime soon, possibly.
Thank you.
Thank you very much for being here.
Thank you very much.
Thank you so much. It was an honor.
It was a pleasure. Thank you.
Please like and subscribe, and don't forget to hit that bell icon.